Imagine looking at a plant, really looking, and seeing not just, a static green thing, but something alive, dynamic,
Right.
constantly talking, fighting, even making friends. That's the incredibly active, hidden world we're diving into today. Welcome to the deep dive. Think of this as your shortcut, your way to really get a handle on the complex ways plants interact with everything around them.
Exactly. We're taking a deep dive into a really fascinating aspect of plant life today. We're exploring how they engage with other living things in their environment. So everything from helpful microbes to attacking insects and pathogens and even competing plants. It's a busy life.
Right. And our mission here is to pull out the absolute most important nuggets of knowledge from our source material. We want to reveal the clever strategies plants use for survival, cooperation, and defense.
Yeah, we'll explore some key physiological processes, look at some pretty surprising molecular mechanisms, maybe touch on developmental stages, and define important ideas along the way. By the end, you should have a really solid grasp of this constant biological dialogue happening, in every garden, every forest. It's everywhere.
Absolutely. It's like uncovering a secret life, isn't it?
Mm.
Okay, let's unpack this. First up, let's talk about the friends plants keep, the beneficial interactions.
Okay, so plants have formed these incredibly long-standing, mutually beneficial relationships with microorganisms. We're talking ancient history here. These partnerships go back hundreds of millions of years. They might have even been crucial for plants first colonizing land. Think about that.
Wow, yeah, we see classic examples of this, right? Like the symbiosis between roots and fungi mycorrhiza, that's ancient. Or maybe the more famous one, legumes and nitrogen-fixing bacteria.
Right. And what's really fascinating is the molecular signaling that controls these associations. How do they talk to each other? For legumes and those nitrogen-fixing bacteria, the rhizobia, it's all about chemical signals called nod factors. These are specific molecules, lipoprotein oligosaccharides, that the bacteria release near the root surface.
Okay, so the nod factors are like a specific chemical handshake the plant recognizes. Is that a good way to think about it?
That's a great analogy. Precisely. The plant has specific receptors, proteins on its root surface, called nod factor receptors, or NFRs. They're a type of receptor kinase with what are called lysine domains. These domains are good at binding these kinds of molecules.
Lysosome domains. Okay,
Yeah.
so they grab onto the nod factor.
Yes, two receptors often work together, NFR1 and NFR5. And when they bind that nod factor, it kicks off two key processes inside the root cells. It facilitates the bacterial infection in a controlled way, of course, and it starts the development of the root nodule.
The nodule, right, that little house for the bacteria where the nitrogen fixing actually happens.
Exactly. And interestingly, there's a common player here, a receptor called SYMRK, which is involved in both the rhizobial symbiosis and the arbuscular mycorrhizal one with fungi.
Oh, that suggests these pathways are related then, evolutionarily speaking.
It really does. It implies that the legume rhizobia system likely evolved by sort of co-opting parts of the older mycorrhizal pathway. They use similar signals too. Fungi use mybiotic factors, also lipoprotein oligosaccharides.
Okay, here's where it gets really interesting for me. I read that a common signal in both these pathways involves calcium spiking inside the root cells.
Yes, it's fascinating. These fluctuations in calcium concentration, these sort of oscillations inside and around the nucleus, act like a critical internal signal. They activate a core set of symbiotic genes. And this, along with cytokinin hormone signaling, leads to the actual development of the nodule.
Wow. And you mentioned the receptors involved are related to defense receptors.
That's right. It suggests that plants cleverly repurposed parts of their defense machinery, originally meant to fight off invaders, to instead recognize and welcome these beneficial partners. A neat evolutionary trick.
That is really smart evolutionary recycling. Okay, what about other helpful microbes? Not the ones in the nodules, but the ones just hanging around the roots.
Ah, you mean the plant growth promoting rhizobacteria? Or PGPR? Good point. These guys live in the rhizosphere, that's the soil zone immediately surrounding the roots. It's a really active area. They basically live off stuff the roots exude, like sugars and amino acids.
So what do they do for the plant in return?
Well, they offer several benefits. They can help the plant get nutrients, like making phosphorus more available. Some can stimulate root branching, helping the plant explore more soil. Some bacteria, like Bacillus subtilis, even release volatile compounds that can change root architecture and enhance iron uptake.
And crucially, protection, right?
And crucially, yes, protection against pathogens.
So they're like tiny microbial bodyguards for the roots, that kind of idea.
In a way, yes. Some PGPR, like certain Pseudomonas species, synthesize antifungal compounds directly. Others don't attack the pathogen themselves, but they trigger the plant's own defense system. This is called induced systemic resistance, or ISR. It basically primes the plant, makes it ready to defend itself more strongly if it gets attacked later. Some bacteria can even produce antibiotics or molecules called siderophores that scavenge iron, making it harder for pathogens to grow. And this production can be controlled by quorum-sensing bacteria sensing their own population density, which plants can sometimes influence.
Wow, complex interactions. Okay, let's flip the script then. Let's talk about the adversaries, the pathogens, and the herbivores.
Right, the harmful interactions. Plants face constant attack from microbial pathogens, fungi, bacteria, viruses, and of course, insect herbivores. And just like the beneficial interactions, this involves intricate defenses from the plant and equally intricate countermeasures from the attackers. It's a classic coevolutionary arms race.
It really is a constant back and forth, isn't it?
Absolutely. The very first line of defense is simply the plant surface. Physical barriers. Think of the waxy cuticle on leaves, or the periderm, the bark, on older stems. It's like skin for the plant.
A physical shield. Makes sense.
Exactly. Beyond that, you have more specialized mechanical defenses. Things like thorns, which are modified branches, think citrus trees, or spines, which are modified leaves, like on a cactus, and prickles, which are just outgrowths of the surface tissue, like on roses.
Those work well against bigger animals, I guess.
They do, but maybe less so against small insects. For insects, other defenses are more important, like trichomes.
Trichomes, those are the plant hairs, right?
Exactly. They can be a physical barrier, just making it hard for insects to walk or feed. But many are also chemical factories. Glandular trichomes store secondary metabolites, things like terpenoids or phenolics, and release them when an insect touches or damages them.
The stinging nettle, that's a perfect and very painful example of this.
It really is. Its stinging hairs are highly specialized trichomes. They're needle-like, reinforced with silica to make them stiff, and filled with a cocktail of irritants, histamine, formic acid, serotonin. The tip breaks off easily, injecting the mix like a tiny hypodermic syringe. It's a very effective mechanical and chemical deterrent. Trichomes can even act as sensors, signaling for other defenses.
Ouch. Plants don't mess around.
They really don't. Another mechanical defense is embedding mineral crystals in their tissues. Grasses use phytoliths, tiny silica crystals, which makes their leaves tough and abrasive, hard for insects to chew. Other plants use calcium oxalate crystals. Sometimes these are sharp, needle-like bundles called raphides. If you bite into a plant like dumb cane, Divenibacea, these can penetrate tissues, cause irritation, and help toxins get in.
And I love the sensitive plant example, mimosa. That's mechanical too, isn't it?
Yes. That's a different kind of mechanical defense rapid movement. The leaflets fold up incredibly quickly in response to touch or damage. It might startle a herbivore or just make the leaf seem less appealing. It's a fascinating response.
So beyond the physical stuff, there's the chemical warfare.
Indeed. This is where secondary metabolites come in. We need to distinguish these from primary metabolites. Primary metabolites are things essential for basic growth and survival: sugars, amino acids, proteins. They're found in pretty much all plants. Secondary metabolites are different. They're often specific to certain plant groups, not directly involved in basic metabolism, and frequently serve defensive roles. Think alkaloids, terpenoids, phenolics.
And some of these defensive chemicals are the ones that are incredibly important in medicine for humans, right? Or sometimes poisons.
Exactly. Hemlock's poison, cytotoxin, or foxglove's digitoxin, used for heart conditions. These are secondary metabolites evolved for plant defense. But they have powerful effects on animals, including us.
So how does the plant avoid poisoning itself with all these toxins?
Great question. Storage is key. These compounds are often kept safely tucked away, compartmentalized. They might be stored in the central vacuole of the cell, or in specialized structures like resin ducts in conifers, or leticifers, the cells that produce latex, or in those glandular trichomes we mentioned. They're only released when the tissue is damaged.
Like a built-in defense system that's only activated when it's actually needed. A booby trap.
Precisely. Think of pine resin. When the bark is damaged, it oozes out sticky, toxic terpenes and resin acids that can trap or poison insects. And this production can even be ramped up after damage, often regulated by signals like methyl jasmonate. Leticifers produce latex, that milky fluid you see in plants like milkweed or poppies. It coagulates when exposed to air, gumming up insect mouth parts, and it's often packed with toxins.
Like the opium poppy latex, obviously.
Right, containing opiates. Or milkweed latex, which has cardinalides, toxic steroids that affect heart function. They make vertebrates vomit.
But wait, some insects, like monarch caterpillars, they eat milkweed. How do they handle those toxins?
Ah, yes, a fantastic example of coevolution. Monarch caterpillars have evolved tolerance to the cardenolides. Not only do they tolerate them, they sequester them, store them in their own bodies, which makes the caterpillar itself toxic to predators, like birds. Their bright orange color is a warning sign. Don't eat me, I taste bad.
Wow. And other insects do this too.
Yes, other specialists like the milkweed bug and certain aphids have similar adaptations. It's an ongoing arms race. Another clever storage strategy is keeping toxins as non-toxic precursors, often bound to sugars, in the vacuole. They're kept separate from the enzymes that would activate them. Think of the mustard oil bombs in the brassica family, broccoli, mustard, wasabi. They store glucosinolates. When the tissue is damaged, an enzyme called myrosinase mixes with the glucosinolate and hydrolyzes it, creating pungent, toxic isothiocyanates.
That's the characteristic sharp taste, right?
That's exactly it.
Right.
Grasses use a similar system with benzoxazinoids, and then there are cyanogenic glycosides. These are particularly dangerous because when the plant tissue is damaged, they release hydrogen cyanide HCN. Yes, the same cyanide. It's a potent blocker of cellular respiration. Plants like sorghum and cassava produce these. Cassava needs careful preparation to remove the cyanogenic glycosides before eating. It's remarkable how many plants we use for food or medicine contain these compounds because they are defensive chemicals.
So given how effective these toxins are, why don't plants just produce them everywhere all the time? Seems like a good strategy.
It really comes down to cost. Synthesizing these complex molecules takes a lot of energy and resources. The optimal defense hypothesis suggests plants allocate defenses strategically. They invest more in tissues that are most valuable for their survival and reproduction and most vulnerable. Often this means young, developing leaves have higher levels of these constitutive, always-present defenses than older leaves. They have more future potential, you see.
Okay, so constitutive defenses are costly. What's the alternative?
Inducible defenses. These are defenses that are switched on or ramped up after the plant detects an attack. This allows the plant to save resources when there's no immediate threat and respond flexibly.
Okay, but how does a plant know it's being eaten by, say, a caterpillar and not just getting hit by hail or a falling branch? It needs to be specific, right?
Absolutely. It's not just about sensing the wound itself, although that's part of it. Plants recognize specific cues associated with the attacker. These can be molecules from the insect, often found in their saliva or oral secretions. We call these elicitors, or sometimes herbivore-associated molecular patterns, HAM-Bs. They act as specific danger signals, telling the plant, this isn't just random damage, this is a biotic attack.
Like velocitin. I think I read about that one.
Yes, velocitin was one of the first identified. It's a fatty acid amino acid conjugate found in the regurgitant of caterpillars like the beet armyworm. When maize detects velocitin, it strongly enhances the wound response and triggers the release of volatile signals, those smells we'll talk about later. Interestingly, the insect makes velocitin using fatty acids from the plant it's eating.
Wow. And there are others.
Yes. Grasshoppers produce a different class called scalephorins. These also trigger defense responses like jasmonic acid accumulation. But unlike velocitin, these aren't derived from the plant itself, and it gets even more specific. Phloem-feeding insects like aphids, which cause less obvious mechanical damage, tend to trigger different pathways, often involving salicylic acid, which we usually associate more with pathogen defense. Chewing insects mainly trigger jasmonic acid.
Okay, so the plant detects these elicitors. What happens next inside the cell?
Well, some of the very earliest events involve rapid changes in cellular signals. A really common one is that spike in cytosolic calcium concentration, K2 plus CIT.
Calcium again. It seems to pop up everywhere.
It really does. It's a ubiquitous second messenger. This increase in calcium activates various proteins like kinases, leading to phosphorylation cascades and ultimately changes in gene expression, turning defense genes on.
So calcium is like an internal alarm bell inside the cell.
That's a good way to put it. It's complex, and sometimes calcium can even downregulate defenses, but generally a rapid increase is a key early signal near the wound and even systemically. Another important set of players are MAP kinases, mitogen-activated protein kinases. These are crucial signal transducers. In tobacco, for instance, specific MAPKs called WIPK and SIPK are essential for different steps in activating the jasmonic acid pathway in response to herbivory. You need multiple MAPKs working together for a full defense response.
Okay, so calcium, MAP kinases. What's the main hormone pathway for insect defense?
For chewing herbivores, the major signaling pathway is the octadecanoid pathway, which leads to the synthesis of jasmonic acid, or JA. JA levels shoot up rapidly after wounding and eliciting perception, and JA itself induces a huge number of defense genes. If you knock out JA production, plants become incredibly susceptible to insects.
And if you add JA back,
You can often restore resistance. It's critical defense hormone. It's synthesized through a series of steps, starting in the chloroplasts and finishing in peroxisomes.
and how does JA actually work to turn genes on?
It works through a really interesting mechanism involving protein degradation, similar to how hormones like auxin work. JA is often converted into an active form, typically jasmine oil isoleucine, JAIL. This active form then acts like molecular glue. It helps a receptor protein called COI1, which is part of a ubiquitin ligase complex, to bind to repressor proteins called JAZ proteins.
Repressors, so they normally keep the defense genes off.
Exactly. JAZ proteins normally sit on transcription factors like MYC2, preventing them from activating JE-responsive genes. But when JAL brings COI1 and JAZ together, the ubiquitin ligase tags the JAZ protein for destruction by the cell's recycling machinery, the proteasome.
So destroying the repressor lets the activator do its job.
Precisely. Getting rid of JAZ liberates MYC2, which can then turn on the defense genes. And importantly, this whole system is often linked to suppressing growth.
So JA is basically telling the plant, stop growing for a bit, focus resources on fighting back.
That's a key part of the strategy, yes, reallocating resources. And JA doesn't just turn on gene expression. It also triggers the production of defense proteins that directly target the herbivore. Things like alpha amylase inhibitors that mess with starch digestion, or lectins that bind to the gut lining and block nutrient absorption, and especially proteinase inhibitors. These block the digestive enzymes, like trypsin, that herbivores use to break down plant proteins. They really reduce insect growth and development.
And I bet the insects have evolved ways around those too.
Absolutely. Some insects produce proteinases that are resistant to the inhibitors, or they just ramp up production of their normal ones. It's that arms race again.
Okay, so we have elicitors from the insect. Does the plant's own damage create signals too?
Yes, definitely. When cells are damaged, they release endogenous elicitors, often called damage-associated molecular patterns, or DAMPs. Fragments of the plant's own cell wall, like oligogalacturonides, can act as DAMPs. These are also recognized by receptors and trigger defense responses, part of the plant's innate immunity.
And these defenses aren't just happening right where the chewing is, are they? You mentioned systemic responses.
Correct. Herbivore damage triggers systemic defenses throughout the plant, preparing undamaged parts for potential attack. In tomato, a small peptide called cystamin acts as a mobile signal. It's released from damaged cells, travels short distances, binds to receptors, and triggers JA biosynthesis in adjacent cells. Then JA itself, or a related compound, is thought to travel longer distances through the phloem, the plant's vascular tissue, to activate defenses like proteinase inhibitors in distant leaves.
So signals moving through the plant's plumbing.
Exactly. And it's not just chemical signals. Plants also use electrical signaling for rapid long-distance communication during herbivory.
Electrical signals? Like a plant nervous system.
Well, it's analogous in function, but very different mechanism from animal nerves. Damage can induce waves of plasma membrane depolarization changes in electrical potential that spread quickly through the vascular system.
How fast are we talking?
Pretty fast for a plant, up to maybe 9 centimeters per minute in Arabidopsis. These electrical signals correlate with the systemic increase in jasminates. Genes related to glutamate receptors, similar to those in animal nervous systems, seem to be involved. Mutants lacking these GLR genes show reduced electrical signals and weaker distal defense responses.
That is wild. Okay, what about those smells you mentioned, the volatiles?
Ah, yes, herbivore-induced volatile organic compounds, or VOCs. This is another fascinating layer of inducible defense. When a plant is attacked, it releases a complex blend of chemicals into the air. These often include terpenoids, alkaloids, phenolics, and those green leaf volatiles responsible for the smell of cut grass. The blend can be specific to the type of herbivore attacking.
What's the point of releasing these smells? Are they just a byproduct?
Oh no, they have critical ecological functions. First, they can act as an indirect defense by attracting the natural enemies of the herbivore, like parasitic wasps or predatory mites that prey on the insects eating the plant. The smell is like a dinner bell for the plant's allies. The velicitin example in maize attracting wasps is classic.
So the plant calls for bodyguards?
Pretty much. Second, the volatiles can repel other herbivores, maybe preventing females from laying more eggs on an already infested plant.
And third, you mentioned signaling to neighbors.
Yes, this is really cool. These airborne chemicals can act as signals between plants. A plant under attack can essentially warn its neighbors. Exposure to these volatiles can prime the defenses of nearby undamaged plants, so if they get attacked later, their response is faster and stronger. Greenleaf volatiles are particularly effective at this.
So plants eavesdrop on each other's distress calls.
You'd say that. Volatiles can even act as signals within the same plant, communicating between branches that might not be well connected vascularly, like in sagebrush, or trigger other defenses like extrafloral nectar production to attract ants.
Amazing communication. Is there a timing aspect to all this?
Yes, absolutely. Many plant defense responses, especially those involving jasminates and even salicylates, are regulated by the plant's internal circadian clock. Herbivore activity often peaks at certain times of day. Studies show that plants aligning their defenses, like the JA pathway, with the herbivore's feeding rhythm provides much better protection and slows down insect growth more effectively. It's about being ready at the right time.
Oh, okay. That covers insects pretty thoroughly. What about pathogens? Bacteria, fungi, viruses?
Right. Plants are constantly bombarded by potential pathogens, yet most are surprisingly resistant. They lack an adaptive immune system with mobile cells like ours, but they have very effective defenses. These involve recognizing the pathogen, triggering specific immunity pathways, producing antimicrobial compounds, and sometimes even programmed cell death.
How do these tiny pathogens actually get inside the plant in the first place?
They have various strategies. Some secrete enzymes like cutinases or cellulases to directly degrade the plant's cuticle or cell wall and force their way in. Others are more opportunistic, entering through natural openings, the stomata, pores for gas exchange, hydathodes, water pores, or lenticels, pores in bark. And wounds are a major entry point, often created by herbivores, which can also act as vectors, carrying viruses or bacteria from one plant to another. Phloem-feeding insects can deposit pathogens right into the vascular system.
Once inside, what do they do?
Their strategies vary. Necrotrophs kill the plant tissue first and then feed on the dead material. Biotrophs need to keep the host cells alive to feed on them. Hemibiotrophs start as biotrophs and then switch to being necrotrophs later. To help them colonize, pathogens produce effector molecules. These are proteins or other molecules they secrete, often directly into the plant cells using sophisticated secretion systems.
Effectors, what do they do?
Their job is basically to manipulate the plant cell to the pathogen's advantage. They might suppress plant defenses, alter plant metabolism to release nutrients, or change hormone levels. Examples include enzymes that degrade plant structures, toxins like fusicocin that mess with plant cell membranes causing wilting, or even growth regulators like the fungus that causes foolish seedling disease in rice by producing gibberellins, making the rice grow too tall and unstable.
So how do plants fight back against these stealthy invaders? How do they detect them?
They have a sophisticated two-tiered detection system. The first line relies on pattern recognition receptors, or PRRs, located on the plant cell surface. These PRRs recognize conserved molecular patterns that are common to many microbes but absent in plants. These are called microbe-associated molecular patterns, or MMPs. Like what? Things like chitin, a major component of fungal cell walls, or specific peptide fragments from bacterial flagella, like FLHE22. When a PRR detects MMP, it triggers MMP-triggered immunity, or MTI. This is a kind of basal defense response, often sufficient to stop non-adapted pathogens. It involves things like reinforcing the cell wall and producing antimicrobial compounds. Controlling stomatal closure is also key. Detecting FLG22 can make stomata close rapidly, blocking entry.
But successful pathogens can overcome this basal defense.
Yes, adaptive pathogens deliver those effectors inside the host cell, specifically to suppress MTI. This evolutionary move by pathogens led to the evolution of the second layer of plant immunity, effector-triggered immunity, or ETI.
Okay, how does ETI work?
ETI relies on intracellular receptors, usually encoded by resistance R genes. These R proteins are specifically evolved to recognize the presence of particular pathogen effectors inside the cell. Most R proteins belong to a large family called NBS-LRR receptors. They have nucleotide binding site, NBS, and leucine-rich repeat, LRR, domains. This recognition is often highly specific, like a lock and key for a particular pathogen strain's effector.
How do they actually recognize the effectors?
Often indirectly, through something called the guard hypothesis. The R protein doesn't necessarily bind the effector directly. Instead, it guards another plant protein, the guardie, that is the actual target of the pathogen effector. When the effector interacts with or modifies the guardie protein, the R protein detects this change and gets activated, triggering a very strong defense response.
And what does that strong response involve?
It kicks off really fast within minutes. You get rapid ion fluxes across the cell membrane, calcium rushes in, protons come in, potassium goes out. A key event is the oxidative burst. A rapid and massive production of reactive oxygen species, ROS, like hydrogen peroxide. Nitric oxide, NO, is also produced. These molecules, ROS and NO, can be directly toxic to the pathogen, but they also act as critical signaling molecules to activate further defenses. Both are needed for the hypersensitive response.
The hypersensitive response, HR, that sounds dramatic.
It is. The HR is a form of programmed cell death. The plant deliberately kills the cells immediately surrounding the infection site. It sounds drastic, but it's highly effective against biotrophic pathogens, as it walls them off and deprives them of living tissue to feed on, effectively containing the infection.
Like sacrificing a few soldiers to save the army.
Exactly. Other defenses activated during ETI include further reinforcing cell walls with lignin or callus to physically block the pathogen, and producing hydrolytic enzymes like chitinases that attack fungal cell walls, and the production of phytoalexins.
Phytoalexins, what are those?
These are a diverse group of antimicrobial secondary metabolites. Unlike constitutive defenses, phytoalexins are typically undetectable before infection, but accumulate very rapidly at the infection site in response to ETI. Different plant families make different types. Legumes make isoflavonoids. Potatoes and tomatoes make sesquiterpenes. There's strong evidence they play a crucial role in resistance in vivo.
So ETI is a strong local defense. Does it have systemic effects too, like with insects?
Yes, absolutely.
Bro.
A localized ETI response, often involving HR, can trigger systemic acquired resistance, or SAR. SAR develops throughout the plant over several days following an initial infection. It provides broad-spectrum resistance, making the plant more resistant to a subsequent attack by a wide range of pathogens, not just the one that triggered it initially.
How does SAR work? What's the signal?
A key signaling molecule involved in establishing SAR is salicylic acid, SA. SA levels rise dramatically at the initial infection site and moderately increase systemically. However, grafting experiments showed SA itself probably isn't the long-distance mobile signal. Methyl salicylate, a volatile form of SA, is a likely candidate for the mobile signal, but other molecules like azelaic acid and glycerol 3-phosphate were also implicated. It's complex.
And how does the plant respond systemically?
A key player in SA signaling in SAR is a protein called NPR1. It acts as a master regulator. Depending on SA concentration, NPR1 interacts with other proteins to control the expression of a large suite of defense genes, including pathogenesis-related PR proteins, which have antimicrobial activities.
Okay, so SAR is triggered by pathogens. What about that ISR you mentioned earlier, triggered by beneficial microbes? How does that compare?
Induced systemic resistance, ISR, is similar in outcome to SAR-enhanced resistance throughout the plant, but the trigger and signaling pathways are different. ISR is initiated by beneficial microbes like PGPR. Instead of directly activating defenses everywhere, ISR often primes the plant. This means the plant is ready to mount a faster and stronger defense response if it's subsequently attacked by a pathogen or even an insect.
Yeah.
ISR signaling typically relies more on jasmonic acid and another hormone, ethylene, rather than salicylic acid, which is central to SAR, though there can be crosstalk between these pathways.
So beneficial microbes can boost defenses too. Fascinating. Are there other threats besides insects and microbes?
Yes, plants also have to contend with parasitic nematodes and even other plants. Nematodes, or roundworms, are tiny soil animals. Many are parasitic on plant roots and cause huge agricultural losses worldwide. They feed using a sharp mouthpart called a stylet. Some, like cyst nematodes and root knot nematodes, are particularly damaging. They induce the plant root to form specialized feeding structures, giant cells, or a syncytium, essentially hijacking the plant's resources.
And plants can fight back.
They can. Parasitic nematodes also secrete effectors, similar to pathogens, to manipulate the host. And some plants have R genes that recognize these nematode effectors and trigger defense responses, sometimes using the same R genes that provide resistance to microbial pathogens.
Okay, and what about plant versus plant conflict?
Good question. Plants compete intensely for resources like light, water, and nutrients. Sometimes this competition involves chemical warfare, known as allelopathy.
Allelopathy, like chemical warfare between plants.
Precisely. One plant releases secondary metabolites into the environment, usually the soil, that inhibit the growth or germination of neighboring plants. A classic example is spotted knapweed, an invasive weed in North America. It releases a chemical called catechin, which is toxic to many native plants, apparently by triggering oxidative stress and calcium signaling, leading to cell death in susceptible species.
A chemical attack to clear the competition. Clever, if ruthless.
It is. And finally, some plants have taken parasitism to the extreme. They parasitize other plants. We can divide them into hemiparasites and holoparasites. Hemiparasites like mistletoe still photosynthesize, but attach to a host plant to steal water and mineral nutrients. Holoparasites like dodder or broomrape have completely lost the ability to photosynthesize. They rely entirely on their host plant for sugars, water, everything. They're often just stems and flowers lacking leaves and roots.
How do they attach and steal?
They develop a specialized invasive organ called a hostorium. It's kind of like a modified root that penetrates the host plant's tissues, tapping into the xylem for water and minerals, and the phloem for sugars in the case of holoparasites. Some parasitic plant seeds even use volatile chemicals released by potential host roots as cues to germinate and grow towards them.
That's incredible targeting. Do we know how host plants defend against these parasitic plants?
It's less understood than defenses against microbes or insects, but yes, host defense mechanisms are likely involved. Signaling pathways involving jasmonic acid, salicylic acid, and ethylene probably play roles, but it's an active area of research.
Wow. Okay. I had no idea the lives of plants were quite so complex. All these friends, enemies, intricate chemical signals, electrical waves, it completely changes how I think about them.
It really does, doesn't it? It's truly a hidden world of constant interaction, communication, and coevolutionary adaptation. We've covered quite a bit today how plants engage in these mutually beneficial relationships with microbes, how they deploy layers of mechanical and chemical defenses, both constitutive and inducible.
Yeah, and how they activate these complex inducible responses based on really precise recognition of attackers using signals like calcium and hormones like JA and SA.
And the communication.
Yeah.
Communicating internally with chemical and electrical signals and externally with those volatile organic compounds, talking to insects and neighboring plants.
Sure.
And defending against nematodes, competing plants, parasitic plants.
From those tiny nod factors acting like a chemical handshake to call beneficial bacteria, to volatile distress signals alerting the whole neighborhood, and the sophisticated plant immune systems fighting off pathogens with targeted cell death. This deep dive has really shown just how dynamic, responsive, and incredibly sophisticated plants are. They're not passive at all.
Not in the slightest. We've seen how they use everything from simple waxy coatings and sharp spines right up to these complex molecular pathways, specialized resistance genes, all geared towards surviving and thriving in really complex, challenging environments. It's a constant biological negotiation.
It definitely makes you look at the plants in your garden or even just a weed growing in the pavement totally differently. They're not just standing there. They're living, breathing entities engaged in this ongoing saga of cooperation, conflict, signaling.
That's exactly the takeaway. When you start to appreciate this constant, intricate biological dialogue happening all around us and even within the plants themselves.
It really makes you wonder what other hidden complexities, what other conversations are unfolding in the natural world that we haven't even begun to fully understand yet.
That is a great question to ponder. A perfect place to leave it until our next deep dive.